As therapeutic interest in nicotinamide mononucleotide (NMN) continues to grow, particularly for its role in slowing metabolic aging and counteracting obesity, there is a parallel need for reliable biomarkers that can track its biological activity. NMN's most promising mechanisms involve restoration of NAD levels and reactivation of autophagy, a key cellular housekeeping process that declines with age. Because autophagy is intricately involved in lipid metabolism, insulin sensitivity, and mitochondrial health, being able to measure its activity in response to NMN is critical for evaluating efficacy, personalizing dosage, and predicting long-term metabolic outcomes. Identifying a panel of biomarkers that accurately reflect autophagic flux, NAD, dynamics, and systemic metabolic improvements would allow researchers and clinicians to monitor the progression of aging interventions in real time. Fortunately, a growing body of research has begun to map out these indicators across blood, tissue, and imaging platforms, many of which offer direct or surrogate insights into autophagy activation and its metabolic consequences. One of the most direct autophagy-related markers is LC3, microtubule-associated protein 1A/1B, light chain 3, particularly the lipidated form known as LC3-2. The conversion of LC3 to LC3-2 is a hallmark of autophagosome formation. In NMN-treated animal models, increased LC3-2 levels in liver, skeletal muscle, and adipose tissue often correlate with enhanced autophagic activity. In humans, while tissue biopsies are more invasive, peripheral blood mononuclear cells, PBMCs, can provide a non-invasive snapshot of systemic autophagy shifts, where LC3-2 changes are still detectable following NMN supplementation. Another widely used biomarker is P62/SQSDM1, an adaptor protein that is selectively degraded by autophagy. Reduced levels of P62 suggest an increase in autophagic clearance. In aging tissues, elevated P62 reflects stalled or impaired autophagy, and NMN's ability to lower P62 expression or restore its turnover serves as a strong indicator of reactivated autophagic flux. Paired analysis of LC3-2 and P62 offers a dynamic picture. Increased LC3-2 and decreased P62 typically point to an actively recycling autophagy system. Beclin-1, a protein critical for autophagy initiation, also responds to NMN through SIRT1 and AMPK signaling pathways. Elevated expression or phosphorylation of Beclin-1 after NMN therapy suggests restored autophagy signaling, especially in liver and adipose depots undergoing lipid turnover. Importantly, this marker can also help distinguish between autophagy induction and lysosomal degradation dysfunction, a distinction necessary for interpreting metabolic outcomes. Because NMN operates in large part by restoring NAD, it's no surprise that intracellular or plasma NAD levels themselves serve as an essential upstream biomarker. In fact, NAD concentration can serve as a surrogate indicator of potential autophagic capacity. As NAD rises, downstream SIRT1 activity, particularly SIRT1, is reactivated. Thus, SIRT1 expression or activity can serve both as a functional correlate of NMN efficacy and as a modulator of autophagy through FOXO and PGC1A signaling pathways. In clinical settings, measuring NAD and SIRT1 levels in PBMCs or muscle tissue offers accessible, predictive value. Another class of valuable indicators involves lysosomal markers, such as LAMP1/2, lysosome-associated membrane proteins, which reflect the fusion and degradation phases of autophagy. Increased expression or activity of these proteins following NMN treatment signals that the autophagic pathway is not only initiated but also successfully completed, a critical distinction in aging cells where autophagosomes often accumulate without resolution. Enhanced lysosomal function, in combination with restored mitochondrial turnover, often correlates with fat mass reduction and improved insulin sensitivity. Looking beyond cell-intrinsic markers, circulating cytokines and adipokines offer indirect but highly informative readouts of NMN-induced metabolic rejuvenation. For example, reduced IL-6, TNF-A, and MCP1, coupled with increased adiponectin, indicate a shift from inflamed, senescent fat tissue to a healthier adipose phenotype. These changes often accompany improved autophagic clearance of damaged mitochondria, mitophagy, and decreased presence of senescent cells, both of which are triggered by NAD restoration. Metabolomic shifts are also revealing. Increased levels of ketone bodies, like SS-hydroxybutyrate, improved glucose tolerance, and reductions in circulating free fatty acids and triglycerides strongly suggest restored energy homeostasis and lipid mobilization, often downstream of autophagy reactivation. These markers, combined with imaging-based body composition tools, e.g., DEXA scans, allow for non-invasive correlation between molecular activity and whole-body changes in fat and lean mass. FGF21, fibroblast growth factor 21, and BNDF, brain-derived neurotrophic factor, are also emerging as metabolic and neuroendocrine biomarkers that respond to autophagy modulators like NMN. FGF21 increases in response to mitochondrial stress and has been linked to white fat browning and improved insulin sensitivity, both processes enhanced by NMN-driven autophagy. Meanwhile, BDNF expression reflects improved neuronal health and appetite control via hypothalamic autophagy, tying NMN's actions to both metabolic and cognitive aging trajectories. In summary, a composite biomarker panel for monitoring NMN-induced autophagy and its metabolic benefits would likely include LC3-2 and P62, autophagic flux, Beclin-1 and LAMP2, initiation and lysosomal completion, NAD, and SIRT1, upstream activation, adiponectin, and inflammatory cytokines, functional metabolic response, mitochondrial DNA/nuclear DNA ratio, reflecting mitophagy, glucose, ketone, and lipid profiles, systemic energy regulation, body composition metrics, clinical outcomes. Tracking these markers before, during, and after NMN intervention offers a powerful lens into how metabolic aging is being reversed at the cellular level. It also opens the door to precision medicine approaches, where the dose, duration, and combination of NMN with other autophagy enhancers can be tailored to maximize benefit for each individual, especially those facing the dual burden of aging and obesity.